Hey guys, so we're going to go through the first type of question that's going to be on your modified AP exam, and that's the long response. So with this type of question, there's a very particular way or formula almost that they expect you to answer these questions. Going through your work, vast majority of you are fine on content. We need to really focus on how to answer the question, how to use the technology. So that's what we're going to work on today. So what I'm going to do is I'm actually going to use the test questions from yesterday, and I'm going to go through them one by one so that you can see exactly how you need to answer the questions to get your full points. So first things first, let's just, oops, stay on the correct screen. Let's just kind of go through what this type of question is actually going to be. So first of all, this is what they are referring to as interpreting and evaluating experimental results. So what you're going to do is you're actually going to have a case study, an authentic situation where you're going to be looking directly at and purposely at a scientist's body of work, and you're going to have tasks related to that. Now, they think that this question is going to take you about 25 minutes on its own to answer it. So what you're about to see is I gave you a much harder task to see how you could do under pressure because this is important. So this test question is extremely important. So question 1 is extremely important in your overall score because it's going to be 65% of your total grade. The 25 minutes to work on this question does not include an additional 5 minutes that AP plans to give you for uploading your response. So within that 25 minutes, you do not have to try and get everything answered and uploaded. So you'll be able to answer it, then take 5 minutes to upload the question before you're expected to move on to question number 2. So when we look at this type of question, what we're actually looking at is the type of question that has 4 very particular pieces to it that AP expects to see in a very formulaic way so that you answer this to their liking. So for instance, the first part of this question is always going to be where you describe and explain some kind of biological concept. Okay, so you're describing and you're explaining. So when you're writing this down, you need to be sure that your answer does those 2 things. And you'll notice that biological concept, what they're getting at is that content piece that I've been trying to give you guys all year. The second part to this question is always going to be to identify, if I could spell, and justify experimental results. Or I'm sorry, design and procedure. We're not to results yet. So you have to be able to look at what they did and understand it. So that would be the identify part. You have to justify what they did in regards to good science. So the second part of this is really those research projects that you did where you had a research topic and you had to design an entire experiment to answer that question. So that's what this is doing, only instead of doing your own work, you have to work with somebody else's. Part 3 then is going to be to analyze results. So you have to look at the data that whatever science group is doing this on the test, and you have to be able to look at it and say what those results mean. So you've gotten lots of practice of this in class. We just have to put it into practice. Now, the last part is going to be to make and justify predictions. So you're either going to look at the results and an analysis that you did and say, this is what it means and here's how we're going to apply it. Or you have to look at what they're saying is the answer to the experimental question, and you have to either go along with it or not, and you have to justify why you're going along with it or not, okay? So when we're looking at this, we've got those 4 basic parts. So great, what does that actually mean in regards to what you need to do? So another piece to this then is to understand the scoring guidelines that are used. So in part A, this is going to be that first part that we talked about, the describe and explain, and it's going to be worth 1 to 2 points, okay? So when we're looking at this, we can see that describing and explaining a biological concept isn't really deep thinking, and so you're not getting a majority of your points by being able to identify the concept and describe it, though those points matter, so we want to do the best that we can. Part B then is going to be 3 to 4 points every time this kind of question is given. So you can see that if I'm identifying and justifying that design and purpose, then that is starting to gain more points. So if you're able to look at the experiment as a whole and look at the pieces and understand what's being done, then you're going to do very well on the part B portion. Your part C of the question is going to be anywhere from 1 to 3 points. And the reason for that vast spread is it depends on what kind of analysis you have to do on the data. Is it a simple finding an average? Is it looking at a trend? Or do you have to actually apply some statistics? So part C can be worth quite a few points in the end. Part D then is going to be worth the most points. It's going to be 2 to 4 points. And if we look at that, that's easy to see because we're actually taking all of the work of the scientists, all of the work that we've looked at and that analyzed and done everything with, and we're doing something with it. And so a majority of your points are going to be on parts B and D. So if you're looking to do this simply for the fact of getting a good score, your big focus is going to be on part B and D of your first question. Not that A and C aren't important, but those B and D pieces are going to be where you have the opportunity to get the best scores. So let's apply all of this then to our test question that we worked on together. So looking at this, we had here, you have all of this background information that you needed to read that was important so that you understood what was going on. Then we went to the graph. You need to be able to look at the graph and understand what's happening. And so each one of these represents a line of cells, specifically cancer cells. We can see that we are looking at SEPin1, and we are looking at percent of viability of those cells. So the hopes of this experiment, if we read back here, is to see if SEPin1 can inhibit cancer cell growth. So the point of this then is to see what concentrations we can put SEPin1 in the presence of cancer cells to stop the cancer from going further. So besides a data table, or besides the graph, we also have the data table so that you get numbers and you can see different cell lines there. That brings us to part A. So going back to what we remember, part A is going to represent our to describe and explain a biologic concept. So let's see how that plays out with our question. So identify the point in meiosis in which separase cleaves the protein complex that holds sister chromatid pairs together. In normal cells, separase is kept in an inactive state until it's needed. So reading that, sister chromatid pairs would have to be separated when we're going through mitosis, because we need to get those sister chromatids to opposite sides so we can split and have two identical cells. So in my mind, I am saying to myself, this is going to have something to do with mitosis. So I need to identify where that point in mitosis is going to occur. So when do those sister chromatids get pulled apart? And then the second part to this is to explain how the progression of cells past sequential cell sequence checkpoints in the activities of enzymes such as separase is controlled by regulatory proteins. So the first thing that I have to do on question A is I have to answer which point the separase is going to be most active in. And if we go back, remember that that's where we're pulling apart our sister chromatids. So I'm going to answer that separase is going to be most active at the metaphase, anaphase transition. Okay. So going back, that's simply remembering my phases of the cell cycle and determining when those chromatids need to get broken apart. So that's going to happen right before anaphase or at the very beginning of anaphase. So at that transition between metaphase and anaphase, separase is going to be the busiest pulling those sister chromatids apart. That's not the entire answer to the question though, because remember we have to explain. So we've identified our concept. Now we have to explain our concept in regards to how the progression of cells past those checkpoints is regulated by proteins. So what you would say, something along the lines of, so with different combinations of, what is it, cyclines and CDKs, your kinases, cells move at different rates through the cell cycle. You could also put mitosis here, right? So what you're looking at then is now you've explained why separase is going to be important. Because separase, if it is not present, is not going to be breaking those sister chromatids apart. So the regulatory proteins are not going to initiate the mitotic cycle. Okay? So that right there would give us the perfect answer to part A. So now if we go to part B, and it says to identify the independent variable of the experiment graphed in figure 1. Based on the data in figure 1, identify a control that shows SEPN1 rather than something else in the culture medium is inhibiting the growth of the cancer cells. So this is a part B. This is where we are going to identify and justify experimental design. So in this case, my B, my independent variable, and I probably should start it with the, right? So I have a complete sentence. So the independent variable in this case is concentration of SEPN1. So we saw that when we were looking at cancer, when we watched the movie, that we're always looking for the least amount of interference that can get us the most benefit from cancer. And so in this case, we want the least concentration of SEPN1 that's going to actually kill the cancer cell's growth. So that's the first part. The second part of this then asks us what the control is. So the control, and if we go back and look at this graph to be sure that we're looking at the right thing. So I'm changing the SEPin1 concentrations. So here's my independent variable. Your dependent variable is always on the y. So even if in your mind you cannot grasp what's actually happening in this experiment, if you're asked about the variables, you need to remember your good dry mix, right? Dependent, which is your response variable, or your response variable is on the y-axis. Your manipulated or independent variable is always on the x. So now I can go back and I can answer that question easily. So the control is the percent of viable cells, with each culture starting at 100%. So every one of those cultures started with 100% viable cells. So now I've gotten my perfect answer to part B. So part C, we're looking at some sort of analysis of the data. So we're going to have to actually look at the numbers and come up with what the answer is. So based on the data in table 1, describe the general relationship between the sensitivity of cells to SEPin1 and the concentration of separase in the cells. So I know that I'm going to look at how concentrated SEPin1 is in regards to how concentrated separase is. Separase is what we want to have in low concentrations so that our sister chromatids aren't being pulled apart and the cell cycle continues. So remember to always go back and say, okay, what are we looking at? What are we looking at? So in this case, our part A is to describe the concentration of separase in cells to SEPin1. So the greater the concentration of separase in the cells, indicates a higher sensitivity to SEPin1. In that, then we move on to calculate the ratio between SEPin1 required to kill 50% of the cells in a cell line that's most sensitive versus a cell line that's least sensitive. So you have to be able to do math and you have to be able to identify those two different cell lines. So the most sensitive cell line is cell line A, and its concentration of SEPin its ideal concentration of SEPin1 is about 10 microliters. With a 3-to-1 ratio. Then the least sensitive is cell line D at 30. Okay. So now I've looked at the data and I've answered it with numbers. So that is my part C. So now that brings us to part D. And in part D, this is the big thing where we have to actually justify and say whether or not the experiment has valid results. So in this case, the scientists hope to test SEPin1 in clinical trials with human cancer patients. The scientists claim one way to test the efficacy of the treatment is to obtain periodic samples of the cancer cells and determine the percent of cells in metaphase. Okay, so that's their analysis and their conclusion of what needs to happen next to test the efficacy or the efficiency of this treatment. And so now you need to provide reasoning to support this claim. So they're not asking if you agree, they're asking for you to go through and say, this is why this is a good claim. So if the scientists claim is correct, then the rate at which cells are cycling through mitosis will decrease, right? We want to stop the cancer from growing because, so here now is my justification. So this is the first part of this sentence is where I'm restating what the scientists' conclusions are. Now I'm giving my reason because SEPin1 should decrease the amount of separase in cells therefore cells will, or I'm sorry, therefore fewer cells will be in metaphase and this would mean the tumor was no longer growing or growing at a, let's say, lower rate. That would give me my 4 points. Okay. So I hope that this video helps you to understand exactly what we're looking for or what AP is looking for in regards to you demonstrating that you can look at material, you can take a scientific concept that's simple, like the concept of mitosis, and you can expand it and apply it in a new situation. And so really that's what the AP test is. It's not just knowing the facts, it's taking those facts and applying them to some sort of new situation that they're going to be giving you. So I hope this helps you. If you have any questions after this, please let me know. Be sure that you go into AP Central and you look through your responses and see where you could have made better, more clear responses to gain more points. Thank you.